3. Hello and welcome to this BioProcess International Ask the Expert webcast. I'm your host, Leah Rosen, the online editor for BioProcess International. Before we get started, just a couple of notes. This webcast is being recorded and will be made available for replay in the multimedia section of our website. We've muted the audio lines, but we welcome you to type in your questions for our speaker in the question and answer window on your screen. After the presentation, we will begin the question and answer portion, and I will ask our speaker your questions. Your questions in the question and answer window will only be visible to myself and our speaker. So thank you for joining us today. It is now my pleasure to introduce our speaker, Jared Isaac from Cygnus Technologies.
Thank you for the kind introduction. Let me start with a quick introduction to Cygnus Technologies. Cygnus has been a trusted partner for robust and broadly reactive host cell protein ELISAs for over 25 years. From release of the first generic CHO and E. coli host cell protein ELISAs in 1998. We spent much of the early 2000s expanding our menu of generic host cell protein kits to cover all the common expression platforms used for production of therapeutic monoclonal antibodies, proteins, vaccines, and viral vectors. Host cell DNA and other bioprocess impurity assays and novel orthogonal methods such as antibody affinity extraction and mass spec were introduced between 2012 and 2019. Today, we continue to lead the bioprocess impurity analysis field by offering the most advanced orthogonal methods for host cell protein analytics. Today, I will cover the following topics. ELISA antibody coverage is dependent upon detection method. Platform ELISA have broad coverage with cell line variants. AAE-MS is ideally suited for gene therapy coverage analysis. ELISA and LC-MS are confirmatory orthogonal methods. Viral vector manufacturing processes require rigorous analytics, including testing for process-related impurities such as host cell protein, host cell DNA, growth media additives, and enzymes used in viral vector purification processes. Even after multiple purification steps, significant levels of host cell protein impurities can be present. While low levels of most impurities can be inconsequential, patient safety demands that host cell proteins be eliminated or reduced to the lowest levels practical to prevent problems such as adverse immune reactions or other off-target biological effects. Impurities can have significant cost implications in drug development and manufacturing processes and negatively impact stability. By identifying host cell proteins early on, companies can reduce process development costs, better assure product efficacy and safety, and help promising therapies get to market faster. Likewise, regulatory guidelines require testing for cell substrate-related impurities, media and purification additives, as well as adventitious agents through vaccine development to study the candidate vaccine's purity, safety, and efficacy. The FDA issued guidance for the industry on chemistry, manufacturing, control information for human gene therapy investigational new drug applications in January 2020, and it continues to provide additional guidelines for cell and gene therapy products and recombinant vaccines. Antibody affinity extraction, or AAE, was developed in response to the limitations of 2D Western blotting. 2D Western blotting lacks specificity and sensitivity. In this schematic, we have a solid phase support and a chromatography column. The ELISA antibodies are mobilized upon the solid phase support. Then, the host cell protein containing sample is passed over the column. Host cell proteins that are immunoreactive with the ELISA antibodies are captured upon the solid phase support. Non-immunoreactive host cell proteins are washed from the AE column. The immunoreactive host cell proteins are eluted from the AE column and can either be detected by 2D page or LC-MS. This is a workflow of how you can analyze harvest material by antibody affinity extraction to determine antibody coverage. An aliquot of a harvest CCF or cell lysate sample is saved, and the remainder of the sample is passed over the AE column. The immunoreactive host cell proteins are captured with the immobilized polyclonal ELISA antibodies. We then have two options for the analysis of the samples. One option is 2D page with silver stain or fluorescent stain, and option 2 is LC-MS. The results look different based upon the analysis. For option 1, there are gel images and a percent coverage is calculated. In option 2, we have a virtual 2D page image, percent antibody coverage, a list of all the host cell proteins present in the sample, and the immunoreactive host cell proteins with their molecular weight and isoelectric point. We also analyzed drug substance material for co-purified host cell proteins by antibody affinity extraction. An aliquot of a drug substance sample is saved, and the remainder of the sample is run over the AAE column. The immunoreactive host cell proteins are captured with the immobilized polyclonal ELISA antibodies and are eluted from the column. Pre and post AAE samples are analyzed by LCMS analysis, and the results are reported as a list of all identified host cell proteins, the drug substance, the host cell proteins reactive with the ELISA antibodies, the protein molecular weight and isoelectric points, and percent antibody coverage. Here we show a 2D page workflow for host cell protein analysis. We have combined AAE with fast protein liquid chromatography. We then perform sample preparation, isoelectric focusing, 2D page, and silver staining. Gels are imaged and spots are counted using Syngene or Total Lab spot map software with scientist data analysis to calculate antibody coverage range. This is a mass spectrometry workflow for host cell protein analytics. We have AAE in combination with fast protein liquid chromatography and sample preparation. Custom mass spectrometry UHPLC methods are developed for each bioprocess. Scientists analyze the UHPLC chromatograms and mass spectra. Stringent bioinformatic data analysis using Thermo Fisher Proteome Discover software is used to identify high-confident protein identifications to calculate antibody coverage. ELISA polyclonal antibody coverage is dependent upon the detection method. 2D Western blotting typically has a coverage range of 50 to 60%. AAE combined with 2D page typically has a coverage range of 60 to 85%, which is also dependent on the staining methodology. Silver staining has a coverage range of 60 to 80%. Fluorescent staining has a coverage range of 70 to 80%. And Cy3 and Cy5 dyes have a coverage range of 70 to 85%. However, combining AAE and mass spectrometry, we can obtain a coverage range of 70 to 90%. So I've talked about coverage ranges, but how do we determine ELISA polyclonal antibody coverage? We use a lower and upper boundary to obtain an antibody coverage range. The lower boundary is calculated by the post-AAE spots divided by the number of unique spots. The upper boundary is calculated by the post-AAE spots divided by the pre-AAE spots. As you can see, the difference between gel and mass spectrometry-based detection is that we are detecting visual spots that are qualitative representation of proteins and mass spectra which represent quantitative ions. ELISA polyclonal antibody coverage is highly dependent upon detection method. In this example, the same sample was analyzed with Cygnus Technologies antibodies and detected with different detection methodologies. The different methods used were 2D Western blotting, antibody affinity extraction of 2D page, and AAE of mass spectrometry detection. As you can see, in 2D Western blotting and AAE 2D page, the same number of pre-AAE spots were detected. However, in Western blotting, fewer spots were detected compared to AAE 2D page. Furthermore, in AAE mass spec, we see the highest coverage range represented, and we can identify more pre-AAE proteins and a higher number of post-AAE proteins. AAE-MS is a highly sensitive detection method for determining ELISA antibody coverage. The Chromatography and Mass Spectrometry group at Cygnus Technologies has curated proteomic databases for all the cell lines and cell samples listed on this page. In particular, we have created proteomic databases for human-derived cell lines, HEK293, HeLa cells, Vero cells, mouse cell lines, NSO and PG13, SF9 cells, and MDCK cells. When you are looking to choose a new platform or custom ELISA, you have two recommended detection options. It is recommended to use AAE with either 2D page and silver staining or mass spectrometer to determine your antibody coverage for your ELISA assay. What types of coverage ranges can you expect from a platform or custom ELISA? An example of a platform ELISA coverage is the F650S HEK293 ELISA. You can typically expect the polyclonal antibody coverage of 70 to 90%. When choosing to do custom ELISA development, they typically have 80 to 90% antibody coverage. However, these are guidelines, and your specific bioprocess may have different coverage determination. This is an example of a custom ELISA development for HEK293 process with the customer sample. The customer sample had had 1,454 spots in the sample and 1,356 spots in the AAE elution fraction. Based on our assessment, the coverage was 88 to 92%. In this example, the green spots are spots that are matching between the pre-AAE and post-AAE gel. Green spots represent proteins that are reactive with the ELISA antibodies. The red spots represent proteins in the post-AAE that may not have antibody coverage. The blue spots represent spots in post-AAE gel that are enriched by the antibody affinity extraction process. In this example, we have used orthogonal methods, AAE, 2D page, and mass spectrometry to determine ELISA antibody coverage for a HEK293 gene therapy bioprocess. The steps in this process were Step 1, working cell bank, Step 2, harvest supernatant, Step 3, end of protection cells, and Step 4, final drug substance. The F650S ELISA antibody coverage of 60 to 90% was seen for steps 1 through 3. However, in the final gene therapy drug substance, the sample volume was limited, and the total protein concentration was too low for 2D page. Gene therapy final drug substances are an ideal scenario for using mass spectrometry. AAE-MS was performed on the final drug substance, and the antibody coverage of the F650S ELISA antibody was determined to be 80 to 89% with the identification of 235 proteins. AAE-MS is well suited for determining antibody coverage of final gene therapy drug substances. SF9 cells are often used for baculovirus-based AAV production and for antigen protein subunit expression in certain vaccines. This is an example of a custom SF9 ELISA with a customer sample determining antibody coverage by AAE 2D page. The pre-AAE gel had 1,585 spots, and after AAE and in the elution fraction, there were 1,214 spots in the post-AAE 2D page silver stain gel. Based on our assessment, the coverage was between 64 to 78%. This is the platform F975 Vero antibody coverage analysis by AAE 2D page and silver staining. The pre-AAE gel had 1,517 spots, and after AAE, there were 1,193 spots in the AAE elution fraction. Based on our assessment, the coverage of the antibodies was between 62 to 79%. AAE-MS was performed using our platform F975 anti-Vero antibodies for our customer sample. As you can see in the 2D virtual gel, there is a high antibody coverage result of 88% with 1129 pre-AAE proteins and 993 post-AAE proteins. Analysis of the top 40 potential high-risk Vero host cell proteins shows that all the host cell proteins present in the sample have antibody reactivity to those proteins. LC-MS is a confirmatory orthogonal method for ELISA quantification of host cell proteins throughout the downstream purification process. In this example, ELISA and LC-MS were used to quantify host cell proteins through purification steps and all the way to drug substance. The panel on the top shows the quantification of host cell proteins through the complete bioprocess. The panel on the bottom shows a zoomed-in view of removing step 1. As you can see, the quantification correlates well between ELISA and LC-MS in nanograms per mil. Thus, Cygnus Technologies' ELISA and LC-MS methods are well correlated orthogonal methods for confirmatory quantification of host cell proteins and downstream purification processes. Today, I've shown data supporting the following. ELISA antibody coverage is dependent upon detection method. Platform ELISA have broad coverage with cell line variants. AAE mass spec is well suited for antibody coverage of gene therapy drug substances, and LC-MS and ELISA are confirmatory orthogonal methods. I thank you for your time and attention, and I await your questions. Any unanswered questions can be sent to me at the email address on my screen.
Great. Thanks, Jared. The first question is, what is the sample volume requirement for coverage analysis by AAE with 2D page and AAE-MS?
I typically ask for samples of 2 to 5 mg per mil total protein and a minimum volume of 20 mils.
Okay. And do you provide top 40 problematic HCPs for each of the cell lines you analyze?
Yes. We have developed extensive lists of the top 40 proteins in many different cell lines, and this comes from the available literature of host cell proteins, as well as Cygnus Technologies' over 20 years of host cell protein experience.
Okay. If you use AAE2DE, WB2DE is not necessary, correct?
Correct.
Okay. And what is the gel size for 2DE?
It is 8 inches by 10 inches.
Okay. And again, if you want to type in your questions for Jared, we'll try to get through as many questions, and then any questions that we don't get through, we'll pass on to him. He can follow up with you directly. So what about those proteins that are not recognized by anti-HCP antibodies?
When we do a mass spec, we provide lists of all the proteins, and you can identify exactly what proteins that there are not antibody reactivity to. At that point, it would be the customer's decision to do a risk assessment and to determine whether or not to proceed or to do a process-specific ELISA.
Okay. Well, thank you, Jared. And thank you to our audience for joining us. The recorded version of this webcast will be available for on-demand viewing on our website, and as a registered attendee, you'll receive a follow-up email providing you with a direct link. We look forward to having you join us at future BioProcess International Ask the Expert webcasts. Look for those announcements in your inbox. Goodbye.